Welcome to Let's Get to the Marks, going through AS Biology, Paper 1, 2021. Let's get into it. Don't forget, at the start of each question, pause the video, grab a calculator, grab a scrap bit of paper, grab a pen, have a go at the question, and then unpause and watch the walkthrough for that particular question. Excuse me, we'll have some coffee. Question 1. Figure 1 shows part of a DNA molecule. Name the type of bond between the complementary base pairs adjacent nucleotides in a DNA strand. 2 marks. So we've got two different types of bond in the DNA here. So the first one is asking us to name the bonding between the complementary base pairs. So you've got to look at this DNA molecule and think to yourself, where are the complementary base pairs? Well, they're going to be opposite each other. For instance, A and T, adenine and thymine, or guanine and cytosine, G and C, opposite each other. What's the bonding that holds those those opposite base pairs? And that's hydrogen bonding. Okay, it's the strongest of the intermolecular forces, and it's hydrogen bonding. Now, when we talk about adjacent nucleotides in the DNA strand, we're actually looking at the strand. And don't forget those gray strands that are in a double helix going up like that. They contain a phosphate, a deoxyribose sugar, and a nitrogenous base. And how are each of those joined together to form that backbone, the phosphates and the sugars? Well, they're joined together by this bond between a deoxyribose and a phosphate between adjacent nucleotides. That's known as the phosphodiester bond. And so this will get you 2 marks. How did you do? If you don't know it, make a note of it, because anything you didn't know, you need to recall. So you should write that down on revision cards. Anything you do know, you don't need to write down. We've got a quick maths one now. The length of a gene is described as the number of nucleotide base pairs it contains. Use the information in figure 1 to calculate the length of a gene containing 4.38 times 10 to the power of 3 base pairs. So we're going to need to zing back up and see what information, because just knowing that there's 4.38 times 10 to the 3 base pairs isn't going to help us calculate the length of a gene unless we know how long a base is. If we know how long one base is, we just times it by that number. But they don't give us the length of one base. How many bases are they giving us the length of here? 1.7 nanometers. And how many bases are in there? 1, 2, 3, 4, 5. So we need to actually find out how many sections of 5 are within that number. And there's a total of 4.38 times 10 to the 3. We know how long a 5 base section is, so let's divide 4.38 times 10 to the 3 by 5 and then times it by 1.7 nanometers, and that's how you'll get your answer. So if we do 4.38 times 10 to the power of 3 divided by 5, you're going to get your first marking point. So this is your first mark out of the 2 marks. It's 876. So they see that anywhere on your paper, the examiner is going to give you a mark. Now times it by 1.7 nanometers. We know we have 876 blocks of 5 in this string of bases, this gene. So let's times each of those blocks by 1.7 nanometers, and we should get not 1.4, that should be 1,489. And it's like 0.2 nanometers total length for the gene. Round it to 1,489 if you like. You'll still get the marks for 1489.2 because they haven't asked us to round. So there we go. 1.3. Describe 2 differences between the structure of tRNA, that's transfer RNA, and the structure of mRNA, messenger RNA. So mRNA is a straight chain or a linear molecule. Do you remember the shape of tRNA? It's not a straight line. It's actually shaped like a clover, like a leaf. I'll draw a little crusty diagram over here of it. Now, do you remember the 3 little prongs coming down from the bottom of the tRNA? They're actually bases, and they're actually known as the anticodon. And mRNA, which is the straight line at the bottom here, the mRNA actually has the codon. So there's another difference there. So one, we can talk about the shape. mRNA is a straight line. It's a linear shaped molecule. tRNA is in a clover shape. Also, tRNA has an amino acid binding site. You see where I've drawn that crusty little rectangle with the word amino. That has an attachment site to the actual tRNA, to the clover. And tRNA has an anticodon, which is the 3 little things sticking down at the bottom of the clover shape. And mRNA has a codon, and they join by complementary base pairing. They form hydrogen bonds there. Okay, let's zing. 1.4. In a eukaryotic cell, the structure of the mRNA used in translation is different from the structure of pre-mRNA used or produced, by transcription. So you've got to remember transcription and translation. Translation happens later. That's the bit where the mRNA attaches to the ribosome. Transcription happens first. It's where the mRNA comes out, and then it's chopped up, not by scissors, but by enzymes and by other molecules. And we call it pre-mRNA when it first leaves the nuclear membrane through a nuclear pore. And by the time it gets to the ribosome, it's just mRNA. It's not pre-mRNA. What's the difference? There are things called introns, which are non-coding parts of DNA that are removed from the mRNA before it goes to translation. So we want to say pre-mRNA has introns. mRNA does not, because it has been spliced. Right, let's get on to question 2. So question 2, figure 2 shows the structure of a human immunodeficiency virus known as HIV. 2.1, name the structures A and B. So A is where the virus attaches to receptor sites on the cell surface membrane of host cells that it wants to invade, and this is known as an attachment protein. And B is a special container that holds the viral RNA, and it's known as the capsid. Don't get it mixed up with the protein coat. I'm going to label PC here. This is a piece of protein that forms the outside of the virus, and often people might mistake capsid with capsule, which is the slimy mucus layer around bacteria. That is not on a virus. The capsule is capsid. 2.2, describe how HIV is replicated. So this one's worth 4 marks, and we really want to talk about how a virus invades a host cell and replicates itself. So the attachment protein joins to a receptor site on the cell surface membrane of the host cell. It then injects its RNA into the host cell. So first of all, first marking point, attaches to a host cell via attachment protein binding to a receptor site on the cell surface or the cell membrane of the host cell. That's going to get us our first mark. It then injects its RNA into the host cell, and the RNA is converted from RNA, which is single-stranded, into DNA. And how does it do this? It uses an enzyme called reverse transcriptase that converts RNA, viral RNA, into the viral DNA. Now, the viral DNA is then transcribed by the actual host cell. So the host cell then sends its RNA polymerase down and transcribes that viral DNA, and then assembles viral proteins. And these viral proteins are then constructed using a bit of ATP from respiration into new viruses. And those viruses, either if they can burst forth from the cell or just be released through the membrane of the cell when they go on and infect other cells. So that is really how a virus replicates. There's some real A-level keys here. You need to mention reverse transcriptase. You need to mention viral proteins being assembled. You need to mention viral DNA being transcribed by the host cell. So these are all things that this technical terms that you must use in A-level biology to score the marks. All right, let's move on to the next question. We're at the end of this question. Just crusty little diagram, just supposed to show sort of how the enzyme turns the RNA into DNA, and then the viruses are released from the cell once their proteins are assembled. All right, we've got a magnification question, a microscopes question here. So we have Uranema marinum is a single-celled eukaryotic organism. U marinum taken through an optical microscope. So here's a photograph or image. 3.1, explain why it is not possible to determine the identity of the structures labeled X on an optical microscope. Well, we can't really distinguish them as separate structures, and that is because optical microscopes use light, and light, the thickness of a light wave, or the limit to the light wave thickness is 200 nanometers. That's the wavelength of light. And that means anything smaller than 200 nanometers, we can't distinguish as separate objects, so we can't actually identify them. So we say that light microscopes or optical microscopes have a low resolution. You know, small things like ribosomes will just appear like a blur under a light microscope. Okay, 3.2. Marinum cells ingest bacteria and digest them in their cytoplasm. Describe the role of one named organelle in digesting these bacteria. So you just got to name an organelle. So just think about the white blood cells and how our white blood cells digest bacteria. And you're applying your knowledge here. So this is an application question. You should be able to spit this out, the whole phagocytosis, and you just need to talk about that. So lysosomes fuse with the vesicle. Another name for the vesicle is a phagocytic vacuole or a phagosome, which is like a temporary vacuole that forms inside cells. And they then fuse with the membrane of that. They release their lytic enzymes. Now, there's three names for that. You can call them lytic enzymes, hydrolytic enzymes. They're essentially digestive enzymes. You can call them lysosomes or lysozymes, sorry. And those enzymes will digest the bacteria. And there we go. That is really going to get you your 3 marks. So the marks are for saying lysosomes fuse with the vesicle, release the digestive enzymes, or lytic, hydrolytic enzymes, however you want to call them. Now, what else can we talk about? We can talk also about how on the ribosome or the rough endoplasmic reticulum, it's 2 other organelles we could name, we could say that the lytic or digestive or hydrolytic enzymes are constructed on that, and then they're given their 3D shape in the Golgi apparatus, and we can pick up the 3 marks for that. We're talking about the construction of the digestive enzyme. Okay, 3.3, calculate the actual length of the cell shown between Y and Z in figure 3. The magnification of the image is times 900. Give your answer in micrometers and to 2 significant figures. Show your working, 2 marks. So you'd get your ruler out, measure between Y and Z. Obviously, I'm on the PC and it's not A4 size, so I'm just going to give you the length. It's 29 millimeters. Pause the video, grab your calculator, see if you can calculate the actual length of that cell. So what you do here is you'd use your I am a K-knob. No, you'd use your I am equation, which is image size over actual size equals magnification, or whack it in a triangle. And so you want to do the image, which is always the thing you measure with your ruler. You'll never be measuring the actual size, not unless you had some sort of tiny ruler. So it's 29 millimeters over 900. Then we need to realize that the units aren't the same. So I need to convert the millimeters to micrometers. So I times by 1,000, and that makes 29,000 divided by 900. And you should get 32.2 or something. And then you're going to round that because it asks us for 2 sig figures to 32. Okay, moving on to the next part of the question. In large cells of marinum, most mitochondria are found close to the cell surface membrane. In smaller cells, the mitochondria are distributed evenly throughout the cytoplasm. Mitochondria use oxygen during aerobic respiration. Use this information and your knowledge of surface area to volume ratios to suggest an explanation for the position of mitochondria in large marinum cells. So this is slightly more difficult application question. You've got to remember what is the difference between small and big cells when you come to your exchange and transport topic? What's the difference between bigger and smaller organisms? Well, we know that large organisms, or in this case, large cells, have a smaller surface area to volume ratio. Bang, 1 mark. Therefore, they position their mitochondria close to the cell membrane. Why? How's this going to be an advantage? It means there's a shorter diffusion pathway for oxygen to get into the mitochondria, because they're going to have a less efficient transfer of oxygen into them. So they need to put their mitochondria close to the membrane to shorten that diffusion distance, so they can get more rapid diffusion of oxygen into the mitochondria to make up for the fact that they have a small surface area to volume ratio. Whereas small cells have a large surface area to volume ratio. They don't need their mitochondria close to the membrane because there's a short diffusion distance. So you could argue the opposite for the small cells, and that's going to get you 2 marks. Okay, so moving on to question 4. This question's about mitosis in cells. Figure 4 shows the arrangement of the genetic material in a cell during the prophase. We can see there's some dark bits, some light bits, some wiggly bits. It's kind of all over the place. 4.1, describe and explain the arrangement of genetic material shown in figure 4, and it's worth 2 marks. So we might notice that instead of being paired up or being neatly arranged on the spindle fibers or lined up in the middle, like in the metaphase, or pulling away in the anaphase in an orderly fashion. The chromosomes are kind of randomly spread or squiggled about inside the cell. So the chromosomes are arranged randomly. They're not yet attached to the spindle, or probably better to say is there isn't any spindle activity yet. The spindle fibers contract and shorten in order to move the chromosomes about. They attach by their centromeres to the spindle fibers. Well, there's no spindle fiber activity, and also they're short and fat. They've condensed. So that's the reason why we know that is the prophase. 4.2, the diploid number of chromosomes in the body of a cell of an insect species is 4. So that's the diploid number, the normal number in a body cell. Basically, they have 4 chromosomes. Put the tick for the appearance ABCD that shows the chromosomes during the metaphase. So during the metaphase, we know they meet up on the equator. I like the metaphase as the metaphase. Look at the stem of the question. It says mitosis. So you might have been getting confused thinking it's meiosis. This is mitosis. So how are they going to line up? Well, there would have been 4 in the interphase, and then they would have formed X shapes when the DNA doubled during the S phase, the synthesis phase before mitosis. So we would have 4 X's, and then in the metaphase, you'd expect them to meet up. So we kind of have 4 X shapes. So it kind of looks like 8 chromosomes. So it's not really, it's 4, because while they're attached, the 2 sister chromatids, we still call them a chromosome. Right, name the fixed position occupied by a gene on a DNA molecule. So if you think of the chromosomes, things like the alleles for your eye color, they're always found on the exact same chromosome in every human being. And so we call that loci or location, or locus, sorry, not location. Loci is plural, locus singular. 4.4, describe how a gene is a code for the production of a polypeptide. Do not include transcription or translation. So they don't want to hear about RNA polymerase. They don't want to hear about helicase. They don't want to hear about complementary base pairing. So how does a gene code for a polypeptide? Well, bases in your DNA are arranged into 3s that we call triplets or codons, and that's going to get you a mark. So bases arranged into 3s. Each codon or triplet codes for a specific different amino acid, one of the 20 amino acids. The specific order or sequence of those triplets or codons gives us the primary structure of the polypeptide, or the order of amino acids in the polypeptide. And there's your 3 marks. Lovely jubbly. I mean, the mark scheme is super generous on this one. They give you a mark for saying bases and a separate mark for saying triplets. So if you just said bases arranged in triplets, you actually get 2 marks, which is a bit crazy. But that's biology for you. Sometimes the mark schemes are really tough, and sometimes they're a bit more generous on certain questions. Right, we're on to question 5. Describe how the structure of glycogen is related to its function. Okay, well, you can see from my incredible D grade level GCSE art diagram, glycogen is heavily branched. So what does that mean if it's heavily branched? It means that it's more easily hydrolyzed, easier to snap some of these glucose monomers off. Faster hydrolysis means you've got more rapid access to energy. This is a reason why animals can move about, because we store our glucose as glycogen, which is more easily hydrolyzed than starch, and plants can't access their energy as quickly. But also, just like starch, glycogen is insoluble, so it has no osmotic effect. In fact, all the storage molecules have to be insoluble. It doesn't matter which type of question you're doing, whether you're doing amylopectin, amylose, they have to be insoluble. And the reason is, if you store glucose, these little orange circles in a cell, you get something called an osmotic effect. They're going to lower the water potential. That little fork stands for that, or WP, water potential. This means that water will rush in by osmosis, swell the cell up and burst it, and it will explode. So we can't store glucose in cells because it's soluble and it will have an osmotic effect. So all storage molecules are insoluble. So just memorize that, whether you're talking about amylopectin, amylose, or starch, or glycogen. It also contains many glucose monomers, and this goes for all of the other storage molecules, i.e. the carbohydrate storage molecules. So, and of course, glucose is the main substrate used for respiration and the production of ATP. So that's another reason why how its structure is related to its function. It's also, we can say here that it's compact. It's not the most compact storage molecule, but it is pretty compact. And what does that mean if you're compact? That means that if you're compact, you take up less volume and less space. So that's a handy thing, right? Amylopectin is compact as well, and amylose is the most compact with its alpha coiled shape, is the most compact out of all of them because it has no branching. All right, figure 5 shows the primary structure of part of a polypeptide. Each shape represents an amino acid, so the circle, the triangle, the square, they're all different amino acids. Yeah. Identical amino acids have the same shape. Okay, thanks for that. 5.2, name the peptidase that will hydrolyze the bond labeled G. Well, it's called an endopeptidase. Anything that breaks the bonds other than the outer two are known as endo. Endo meaning in, exo means out. So exopeptidases only. Okay, the next question, 5.3. Give the number of different R groups in the polypeptide shown. So look, let's say all the circles have a blue R group because all the circles represent the same amino acid, so they have the identical R group. The triangles are another one, they'll have different R groups, and the squares are a different amino acid. So I've done them in different colors here. They'll have a different R group. So how many different R groups are there in total? And the answer is three different R groups. Remember, the R group is the only thing that differs between the 20 different amino acids. Okay, we've got another little maths question here. So a scientist used an enzyme to digest a polypeptide containing 101 amino acids. The digestion produced a range of smaller polypeptides. The scientist determined the number of amino acids in each of the polypeptides produced. He also counted the number of polypeptides of each varying length. Table 1 shows some of the scientist's results. So we want to circle. As soon as you see something like this, there's 101 amino acids. They've given us that info for a reason. Number of amino acids in the polypeptide. So this first one here has 5 amino acids, and there are 2 of them. And the second type, there are 6 amino acids in this next protein or polypeptide, and we don't know the amount. And then there's 15. I'm not going to draw that. That would take too long. And 20. So we know that we've got 2 lots of 5 and 3 lots of 15. So let's work out the total amount of amino acids that we do know. So we've got 5 times 2. That's 10 amino acids in the first 2 polypeptides, and then we know we've got 3 polypeptides that are 15 amino acids long, so that's 45. Add them together, we know that we have 55 amino acids accounted for. So let's do 101 minus 55. That gives us 46 amino acids left, and we know the length of the chains can be either 6 or 20. So what do you reckon the combination is to make 46 if you have 20 and 6? We must have 2 20s and 1 6. There's no other way to put those together to make 46. So we've got 1 peptide, 6 amino acids, and 2 that are 20 each. And that's the end of the question. Okay, moving on to 6.1. First of all, I want to take this time to say it will be a massive help if you can just take your time to hit the like. It massively helps me out.
If you push the button.
Would you like it?
Like.
Big thanks to everybody who hit the like button. Give the pathway a red blood cell takes when traveling in the human circulatory system from the kidneys to the lungs. Do not include descriptions of pressure changes in the heart or the role of valves. So don't start talking about contraction of the atrium, ventricle, and the valves, and this valve opening and that valve closing. They just want you to describe the journey. That's an ear. That should be a kidney. From the kidney to the lungs. So this is more like a kidney. That looks better. Kind of, yeah. All right. D grade, GCSE. So what? Here we go. So what you got to say to yourself is, what vein is going to take the blood from the kidney to an even bigger vein that's going to deliver the blood back to the heart? Which section of the heart is that blood going to enter? And then from there, which blood vessel will it leave the heart via in order to get to the lungs? And if you can name all of those, you can get 3 lovely marks. So we're looking at deoxygenated blood leaving the kidney via the renal vein. And then from the renal vein, it travels to the vena cava, into the right atrium, down into the right ventricle, and then from the right ventricle up through the pulmonary artery. Now, the pulmonary artery is the only artery carrying deoxygenated blood, and we always draw it as blue. It's not really blue in real life, but we always draw it as blue in diagrams. So deoxygenated blood to the lungs. I know those lungs look like testicles, but they're actually supposed to be a pair of lungs. Again, that's down to my poor art grade. Right, figure 6. 6.2. Identify the type of blood vessel labeled M in figure 6. Explain your answer. The type of blood vessel. So we've got two blood vessels there. You can see they've both got thick walls. M has a thinner wall than the one next to it. So you can probably guess what the one next to it is. And it has a larger lumen. Which blood vessel has the thinner walls and the larger lumen? It's a vein. They don't need as much muscle or elastic because the blood is at lower pressure. They can have a larger lumen. Blood's all at lower pressure. With the artery, they need the thick wall, lots of elastic to deal with recoil. Okay, anywho, let's move on. 6.3. Tissue fluid is formed from blood at the arterial end of a capillary bed. Explain how water from the tissue fluid is returned to the circulatory system. Okay, so your blood leaves the heart via the left ventricle. Then it's going to go down an artery and it's going to break off onto smaller blood vessels, still like small arteries, called arterioles. And then it's going to go through a capillary. Obviously, I've drawn little holes in the walls. They're supposed to be the fenestrations. They're like gaps in the capillary wall. And then it gets to the venular end where it will connect to a venule and eventually back to a vein and back to the heart. So as the blood comes down, it contains red blood cells, plasma proteins, and all the things you'd expect in the blood, water, glucose, and that kind of stuff. Now, the high blood pressure created by the left ventricle is going to force small molecules out on the arterial side. But this question is asking us, how does water return from the tissue fluid to the circulatory system? So we're not really dealing with the arterial side. Obviously, water is squeezed out here, shown by this blue arrow. Small water molecules, there's osmosis going on, but there's also the high hydrostatic pressure caused by the left ventricle. But on the venular end, the water potential is higher in the tissue fluid than in the blood, so we get osmosis from the tissue fluid back into the capillary in the blood. Now, why is suddenly the water potential higher? Well, we lost some water at the arterial end, and also the plasma proteins get left behind. And because water's been lost and the plasma proteins are too big to get through the fenestrations or the small gaps in the capillary wall, this means that the blood in the capillary, its water potential lowers so that it's considerably lower than the water potential of the tissue fluid. So water moves back into the capillary from the tissue fluid. Water also moves into lymph vessels, and lymph vessels work by, as you move about, the contraction of your muscles. So more of a pressure system that forces water back into the circulatory system and fluid back into the circulatory system as well. So you're kind of going to get the marks for saying plasma proteins too big, they stay behind in the blood, they lower the water potential in the blood of the capillary, water moves in from the tissue fluid back into the capillary at the venular end and by osmosis. And that's going to grab you all those marks. Right, let's go to question 7. Let's get this into focus. I suggest you pause the video and read through the stem of this question. I'm just going to get down to how to answer the question because reading through all of that is quite a big thing. But you should pause the video and have a little scan read. If you're doing the paper, you printed it out, then highlight key points and let's have a little look. Essentially, they're comparing two plots in a field, two equally sized plots, and they're mowing the lawn. And after 10 weeks, seeing if the field, the plot 2, which had the cut grass, has got a greater diversity than the plot 1. So what's the effect? So here we go, 7.1. Use the information in figure 7 to calculate the index of diversity for the insects captured in plot 1. The formula to calculate the index of diversity is here, okay, where capital N is the total number of insects and little n is the number of insects of one species. Give your answer to two significant figures, and I'm going to do my working out here by the graph. So we're only looking at the data from plot 1, and what I need to do here is work out the sizes. So So what I need to do here is write out the total number of species of individuals of all the species, which is 79, and multiply it by 79 minus 1, which is 78. Now I want to divide it by the total number of each. So the total number of 79 times 78 is 6,162. Now I want to sum the individual total of each species minus 1. So this top one here has got 2 species in it. So in each square is worth 2, and that's a 1 square thick bar. So that's 2 times 1. So 2 times, because we want to do 2 times 2 minus 1, which is the same as saying 2 times 1. So that's 2. The next one, I want to go up here and work out what this is, bearing in mind each square is 1. That's 2.5 squares. So that's 63. And then that goes up to that bar, which is 77. So 77 minus 63 is 14. So I want to do 14 minus 14 times 14 minus 1. So that's 14 times 13. My next bar starts here at 22 and ends at 63. So that's 41 in between. So I want to do 41 times 41 minus 1, which is 40. So I want to do 41 times 40. Notice if you don't put these all in your brackets, you're going to get the wrong answers. And I know it's laborious, but it's easy marks. And then the last section is 22. So 22 times 21, because 22 minus 1 is 21. And these are represented by the lowercase times the lowercase n minus 1. So now I want to do 6,162 divided by the sum of all of that down there. And that is going to give me my index of diversity. Now remember, one of the marks was for putting to 2 significant figures. So if we forget that, we're going to only get 1 mark, even after doing all of this hard work. So final answer, when I add all of this stuff up, oh, I'll put a 1 there. That's supposed to be a 2. Sorry, I don't know how I did that. The final answer is 6,162 divided by 2,286. And that's going to equal 2.7, I don't know, 2.72, 2.73. But remember, we want to go to 2 significant figures. So on my calculator, I got 2.695. And so I'm going to round that up because the 5 is bigger, so 70, and then leave it as 2.7. And that is the 2 significant figures. So there we go. That is the index of diversity. That gives us an idea of the biodiversity of plot 1. Okay, let's zing down. Now, they don't ask for you to do the biodiversity of plot 2, but I reckon it's going to be less because the species evenness was worse. The student concluded that the cutting the plants with a lawnmower increased the species richness of the insects in the meadow. So they've basically looked at plot 2 and said, oh, look, it's increased the species richness. No, it hasn't. There was no difference in the graph with the number of species. Both plots had 4 species. Species richness tells you the number of different species, and in both plots, they still had 4 species. What had actually changed over time was the abundance, the amount of each species. So there were a lot more, whatever that top one was, leaf beetles or whatever, in plot 2. So the abundance changed, but the number of different species remained the same. There was 4 different species in each plot. And so actually the species richness did not change, so the student was not correct. 7.3, the student wanted to use the data from plot 1 to estimate the total number of beetle species in the meadow. Suggest how the student should use the data from plot 1 and the other information provided to estimate the total number of species in the whole field or the whole meadow. First thing to do is calculate the area of the plot. So you do width times height or whatever. Get the area of the plot in meters squared. Remember, the plot is just a small part of the meadow. So here might be my meadow, lovely jubbly, lovely field. And then here is the plot that I've analyzed. Maybe that's 2 meters squared. And the whole meadow is 50 meters squared. The next is to find out, well, how many plots would fit in 50 meters? Say there's 7 creatures or organisms in my 2 meter squared. Then I need to do 50 divided by 2 times 7. Because I can fit 25 plots in there. And if there's 7 on average in one plot, I would do 7 times 25. And it saves me having to count the whole meadow. So here are the instructions for how you do that. And each one of these would score you a mark to get you 4 marks. Boom. Right, moving on. Question 8. A student investigated osmosis in chicken eggs. She dissolved the shells of the 2 eggs without damaging the cell content inside the shells. She then measured the mass of each egg without its shell, covered one egg with vinegar, and covered the other egg with a sugar solution. Kept both eggs covered at 30 degrees for 24 hours. After 24 hours, she measured the mass of each egg. The student designed table 2 and added her results in the table. So just one improvement to the design of table 2 and one improvement to the way she presented the data in table 2. So what's one improvement? Well, what she hasn't included here is what she changed, called the independent variable. And you would normally put the independent variable in the first column. So she said that she changed vinegar or sugar. So the name of the solution covering the egg. She's put that in the third column. So the independent variable, whether it's vinegar or sugar, should have been in the first column. So that was a bit of a big boo-boo. We always put the independent variable in the first column. Now look at how she's presented the data badly here. She said the ratio with the vinegar was 1.29 to 1, and then with the sugar 0.7 to 1. She should have said 0.70 to 1 in order to make sure that the decimal places are identical in her data. So if she's going to 2DP, you've got to do it for all your data. So yeah, that is the way she badly presented her data. She should use the same number of decimal places for all of her data in the final column there. And that would be a better way to present it. And there we go. Let's move on to the next part of the question. Okay, where are we? 8.2. Suggest and explain the advantage of carrying out the investigation at 30 rather than 20. Pause the video, have a go. Okay, think about 30 degrees. What's better at 30 degrees? You've got more kinetic energy. Particles are going to move faster. We're not using enzymes this time. We're using particles because this is an osmosis practical essentially. So if the particles are moving faster, osmosis will happen more rapidly. Our experiment's going to get done more quickly. We're going to get a bigger difference in the same time in the mass of the egg. So if you're using it at a higher temperature, more water is going to get, if you're doing it over 24 hours, which they were doing in this experiment, you're going to get a bigger difference in the mass of the egg, and therefore your results are going to be more accurate, smaller level of percentage error. So 8.3, the student concluded from the information in table 2, the water potential of the solution inside the egg is higher than the water potential with the vinegar. Well, let's have a look at the water potential in the egg compared to the vinegar. Well, it was 66 grams, the mass of the egg, and then after it had been left in the vinegar, it went up to 85 grams. So what does that tell us about the relative water potential inside the egg and inside the vinegar? So what we have to think is, if the mass of the egg has increased, what does that tell us? So the mass of the egg has increased. What has moved into the egg from outside? So here's my egg here. And after it was left in vinegar, it swelled up. So that tells me that water must have moved in by osmosis, and water must have moved in from a higher water potential to a lower water potential. So the student is actually wrong, because the student said the water potential inside the egg was higher. The water potential inside the egg must be lower, and the water potential in the solution must be higher, because water moves down a water potential gradient by osmosis. So there's your 3 marks. Now let's move on to the next question. So 8.4, the student wanted to determine the water potential of the chicken eggs. She produced a dilution series or sugar solution, followed the procedure described on page 20. She calculated the final mass to initial mass ratio of the egg covered in each sugar solution. How would you advise the student to use her calculated ratios to determine the water potential of the eggs? In your answer, state the independent variable in the student's investigation. So what she's measuring and calculating is the ratio of initial mass to final mass for each egg. That way it's easier to compare. It's like using percentage change in mass. So what she needed to do was create a different dilution, a dilution series of different sugar concentrations, and she should plot a calibration curve. Now, the sugar concentration is the independent variable here. That's what she's changing, but she should plot a calibration curve using known concentrations of sugar solution. Now she just needs to plot her ratios, and where the ratio crosses the line or the line intersects the x-axis at 1 for the ratio, that is going to be where there is no net movement of water into or out of the egg, because the initial mass and final mass have a ratio of 1, which means that it hasn't got bigger or smaller, the mass hasn't changed of the egg, and that means that there's been no net movement of water in or out, and it means that you know that that egg is in the same concentration solution outside. The external solution is the same as the concentration inside. So we know that the water potentials are therefore the same. And we can convert also if we want to find the water potential, we need to convert the concentration into water potential, i.e. by looking up, if you have a 0.2 mole per decimeter sucrose solution, what is the water potential of that? So once you know the concentration, you can convert it to water potential. Okay, final question, peeps. Read the following passage. Kidney cells produce a glycoprotein hormone called erythropoietin, or erythropoietin, whatever you want to say. An EPO molecule contains 165 amino acids and approximately 60% of the mass of a carbohydrate. The EPO is transported in the blood, stimulates the bone marrow to produce red cells. In this way, the red blood cells are produced to maintain the blood's oxygen carrying capacity. Some athletes are basically cheaters, and they use EPO by injecting it into their body. The athletic authorities have a program of drug testing to detect the athletes who have injected EPO. In this program, an ELISA test is administered. That's the enzyme linked test. So what we're going to look here is 9.1. Kidney cells produce a glycoprotein called erythropoietin, EPO. Identify 2 organelles in the kidney that enable the production of EPO. So what can make proteins? What would you make? What organelles would make proteins? Well, you've got the ribosome, which is going to, or the rough endoplasmic reticulum, which is going to construct the protein. It's where translation occurs, and then it folds inside the rough endoplasmic reticulum. But in the Golgi apparatus or Golgi body, we package and modify that. So those are the 2 organelles you want to mention. Explain the biological advantage to athletes of injecting EPO. Well, if you inject EPO, it told us that you produce more red blood cells from your bone marrow. If you have more red blood cells, you have more hemoglobin, which means you can pick up a greater volume of oxygen, and therefore you can do a higher rate of respiration, more ATP produced, more muscle contraction, more ability to do aerobic exercise for longer and not use use lactate fermentation, i.e. anaerobic respiration, which produces lactic acid and tires or fatigues the muscles. So any 2 of all of that. 9.3. Describe how mice injected with EPO produce an anti-human EPO antibody. Line 14. So how does injecting mice with human EPO produce an anti-human EPO antibody? So you've got to think, you've got phagocytosis occurring in the mice. And during phagocytosis, the white blood cells of the mice engulf the human EPO that's been injected to them. They see it as a foreign invading particle, and they recognize the antigen as foreign. They engulf it, they digest it, and then they present the antigens on their body. They become APCs. Now, the mice also, just like humans, have T helper cells, THCs, and these have specific binding sites or protein receptor sites that bind to the antigen, specific one, and that's when you have the whole clonal selection on the APC, and then that T helper cell will stimulate B cells to differentiate, to clone themselves and divide into like memory cells and plasma cells. And those plasma cells will make an antibody that is complementary to that human EPO antigen. And that's how we make monoclonal antibodies. We can now take that B cell. I mean, actually, really, they take that B cell, don't they? They make a hybridoma by cloning it with a tumor cell just so they can make lots and lots and lots of copies of the monoclonal antibody. But it's not asking us about that. So that's all your mark. So during the ELISA test, what is the role of the anti-human EPO antibody? Well, that is to bind to the EPO antigen. Now, this is part of what we call the indirect ELISA test. This is slightly more complicated than the direct ELISA test. I've got a video you can check out here with a question, which will go through it, and probably better than I'm about to do it now. So to bind to any antigen or EPO in the sample in the well. What is the role of the anti-mouse antibody with the enzyme attached? Well, this antibody actually binds to the antibody that's already attached to the antigen. So this antibody is complementary to the human antibody, the human anti-human antibody, and that binds to that. And it also contains an enzyme which will change color with a substrate. Okay, so just to explain here, what I'm doing is I'm drawing the antigens. So they bind these, immobilize these to the bottom of a plastic well. Okay, so like the bottom of a container, these are just immobilized and stuck there. Then they put the sample in there of the athlete's blood. Now, if it contains the anti-human antibody, it's going to bind to the antigen, and we know those are little cheats, but we need to prove it. We need a color change. So what we do is we add a second antibody. Now, this antibody recognizes the first antibody and attaches to that, and this antibody has an enzyme linked to it. Okay, now the green antibody with the enzyme linked to it does not attach to the antigen. It will only attach to the antibody that the human has for the EPO, so which proves that they've got EPO in their system. So it's really important that you do washings with the ELISA test as well, because when you add the substrate, if you previously have washed it and the person wasn't guilty, they had a negative result, then there wouldn't be any of the antibody in them, and then you wouldn't get a color change, and you know that that's negative. So what happens here is this person is a cheat. So when we add the substrate, you're going to get a color change because that enzyme linked antibody, the green one, will not have washed out when you wash it with water. It will have stuck there because it will be stuck to the pink antibody that's stuck to the antigen at the bottom. However, if we had this situation, have a look here. This person is a not guilty athlete. Therefore, when you wash that out, the second enzyme linked antibody will not be present. And so when you add the substrate, the enzyme's not present, so you don't get a color change. That's a negative result. They haven't been cheating with EPO. And if that makes any sense, you are a genius. Anyway, these are your answers here. 9.5. Some people object to the use of monoclonal antibodies in testing programs. Use the information. Well, why are they going to object? Because they tested it on, well, they used mice to produce the antibodies and they injected it into goat. Sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.